It's now my great pleasure to introduce this year's winner of the Canada Gairdner Whiteman Award. And it's a great pleasure because the awardee this year, Connie Eaves, and I first met, I think it was about 50 years ago, in Jim Till's lab. And I think it does speak to also the importance of critical mass and the importance and the contributions that Canada has made, Canadian scientists have made to the stem cell story, starting with Till and McCullough, of course, and continuing on to lots of people. I won't name them all because I'll forget some, but including this year's Whiteman awardee. Connie is being recognized for her pioneering work and leadership in the study of hematopoietic, mammary, and cancer stem cells, and her dedicated advocacy for early career investigators and women in science. And indeed, Connie has been a builder. She has really demonstrated outstanding national and international leadership. She co-founded the Terry Fox Labs in British Columbia Cancer Agency, was a leader in the Canadian Stem Cell Network. She held multiple senior positions at the old National Cancer Institute of Canada, where she spearheaded the establishment of the Canadian Breast Cancer Research Alliance, and she has also really strongly advocated for women in science. It's also a pleasure to see sitting beside her here in the audience her husband, because behind every great woman is a man, Alan Eaves, who has worked closely with Connie both as a partner in life but also a partner in science. Connie Eaves.
So thank you, Alan, for this wonderful introduction, and welcome to a change in thinking from molecules, the tinker toys that, you know, guys like to play with and understand because they're so finite and wonderful, to the world of cells. So I just remind you of Ron's analogy with roads and cars going down roads. I think cells are like cities or countries. They're just infinitely more complicated. And eventually we'll understand them all in terms of the molecules in them. But it's interesting, you know, you can understand how to use a road to move stuff from one place to another, but I don't think you can predict accidents yet. So I started off even as an undergraduate, interested in how things grow and develop. And early on, I wanted to be a doctor and treat people. And so I was also interested in cancer. So that is the big overarching problem. We've heard people alluding to it today. We know what it is when you go and get a diagnosis. But actually, if you want to really study it, it's not so easy to know exactly what it is. And we're really interested in understanding how it actually gets started, because most people think that prevention and real treatment is going to involve getting at the very earliest events, because those are the ones that probably most cells in the malignancy still have. Nevertheless, it's pretty clear from even analyzing fairly crudely that there's something goes wrong with how proliferation is regulated, and there's something that goes wrong with how differentiation is regulated. And if you're going to understand that, you have to know something about the normal tissue. So what are the sort of prevailing ideas? You've heard them already. Cancer is caused by mutations, and cancer presumably, or has been historically thought to be largely initiated in primitive cells that have the amount of proliferative activity that allows them to accumulate rare mutations in spite of all this wonderful protection mechanism for preventing that. So yes, back in 1970, I came to the OCI, and Jim Till was kind enough, brave enough to take on what I understand is the first female postdoc ever at the Ontario Cancer Institute that lasted more than a week or two, and work on blood. Why blood? Because in those days it was really the only accessible tissue to really analyze, and the disease, the cancer, leukemia, had a poor outcome. In fact, to this day, it still has a poor outcome, so it's still worth working on. And as you'll hear in a minute, there was some wonderful things happening in Toronto, but really not very much was known about very primitive cells. Well, the idea of how tissues develop from primitive cells, even from stem cells, was not a new idea. The tool of the time was the microscope, and the microscope had looked at tissues, and you could actually see, what happened to the pointer? You could actually see that there must be cells that give rise to the differentiated cells that disappear that don't disappear. So there must be a cell that can self-sustain. And in the middle, they get amplified and they differentiate. And that, even before my time, stimulated Waddington to create this model, which you'll see in many slides now. Is still a prevailing notion, although it's clear it's oversimplified. And Shinya Yamanaka showed that actually you can go back up here, which we know happens every time a germ cell is made and redevelops. But this idea was not easy to investigate in blood, because blood is made in the bone marrow, and bone marrow is the one tissue where you don't have this nice organization. You can't really tell what's going on. And so it was pretty obvious that we needed a different strategy. The world needed a different strategy. And that is, in fact, where Jim and Bunn came in, because it had been discovered in the 50s in Britain that you could take a lethally irradiated mouse and you could reconstitute the destroyed hematopoietic blood-forming system by transplanting in cells from another mouse, healthy cells. And so it was clear there must be a cell that could reconstitute the blood-forming system permanently in mice. And everybody who was interested in that problem all over the world was trying to work on what is that cell. And Jim was a physicist. Bunny was a physician. So Jim thought mathematically about things and said, let's do a limiting dilution assay, and we'll get down to where sometimes it happens and sometimes it doesn't. And then that'll be when we're at a stage where we have close to purified cells, or at least cells we can quantify and analyze. And my understanding is that when they were doing those experiments, Bunny came in and looked at the dead mice, because all good physicians think about autopsies, and he discovered that the spleens of the mice had these little bumps on them, and they together made the seminal leap that they might be colonies derived from single cells, because colonies was a big buzz at the time. And then they, of course, had a whole bunch of very talented students and postdocs who, under their direction, showed that, yes, they were colonies. And many of the colonies had all of the different blood cell lineages, so we knew for sure then that there was a cell that could give rise to all of the lineages. And not only that, each of these colonies, which they proved came from single cells, also variably contained cells that, if you took them out and did the whole exercise again, would make new spleen colonies. So these folk and their groups introduced these very powerful concepts. I won't read them all because of time, but the whole idea of using rigor and quantitation in a field that had been governed by microscopy and descriptive work was revolutionary. And I was addicted to that to this day. So what came next? Well, the idea was that let's fill in the blanks in between the spleen colony forming cell and the cells that you can recognize morphologically. And let's do something that you could do with human cells, because they're not going to put human cells in a mouse. And so there was an era of development of colony assays. The Israelis and the Australians, Leo Sachs's group and Don Metcalf, led the development of developing colonies of granulocytes and macrophages. And actually Arthur Axelrad and his group in Toronto developed an assay for this cell for the mouse. These happen to be human colonies because they're a little nicer and bigger. And then this one, and I came along and showed that there was actually an intermediate cell, which is a different cell, and then we went on to show that these are actually all derived from different cells with different properties and are regulated differently. But this all happens in a couple of weeks. And then the cells are gone. They've all differentiated out. So there must be something earlier. So our group set about trying to figure out that, and we exploited an observation that had been developed by a colleague in Manchester, Mike Dexter, with my former PhD supervisor, Laszlo Lida, showing that if you actually put mouse cells, mouse bone marrow cells, or onto stroma, you could generate hematopoiesis for a long period of time. So we thought, oh, maybe if we do that and then use this as the readout, we will get to the point in time where these cells are still making these cells, but these cells don't exist anymore, the ones that were in the original population. And in fact, it turned out you could separate them away, and you could, with a minimum of 6 weeks, get a very sensitive and quantitative assay. And we turned this into a limiting dilution assay, first pioneered by Heather Sutherland, who was MD PhD student at the time, now still active on the bone marrow transplant program in Vancouver, which Alan, of course, immediately took all the best people and employed them. So then what next? Well, we learned in the field that spleen colony forming cells are actually not the most primitive cell that self-sustain long-term. They kind of last a few weeks in a mouse, their progeny lasts, but they don't sustain more than a few generations. And there are other kinds of cells that last longer and longer. And there are, in fact, cells that will permanently reconstitute a mouse for its lifetime. So we thought, you know, we're assay builders. We'll design an assay that quantifies those cells, because you need to be able to quantify them, to purify them, to understand them, and to understand how they're regulated. So Steve Salvasi, PhD student back in the late 80s, took this on as a project. And we used the principles of limiting dilution, but now we had to have a system where the mouse, where we were going to read out the progeny of single cells, had to stay alive. It couldn't die like Jim and Bunny's assay. Turns out that you can fill them up or have them have lots of short-term repopulating cells, so they get over the immediate problem of dying because they don't have blood cells. And they can have a very small number of the long-term repopulating cells, which allows the assay still to be very, very sensitive and specific to pick up incoming cells that can be genetically distinguished. So that became a beautiful assay, and until these cells were purified to homogeneity a few years ago, it was the gold standard and is still often used in many circumstances. So then fast forward to the late 1980s, John Dick here in Toronto, who Alan didn't want to mention because he might forget somebody, and Irv Weissman's group in California simultaneously discovered that if you took the immunodeficient mice that Jackson Lab, Lenny Schultz in Jackson Lab was creating genetically over time, getting better and better, more and more immunodeficient mice, but still the very first ones, you could stick a human cell into the vein of that mouse. If it had repopulating activity, it would zoom into the marrow and make progeny. And actually the first ones that were found were these guys, and then later these guys, and more recently these guys. This is just the people in our lab who helped contribute to this. A whole field has worked on this, although John has probably been the major player. So now fast forward also, flow cytometry has come along, just like the methods for purifying proteins. Flow cytometry is a method for purifying cells, and you can purify them viably, so you can still do functional assays on the cells that you isolate. You don't need to pay any attention to all this stuff, just to say that this is what the field has thought recreates the Waddington plot way up there in the morphologically unrecognizable component of early blood cells in the human system. And John Dick's group was first to publish that you could isolate cells that would reconstitute mice long-term, human cells reconstitute to immunodeficient mice long-term at 10% purity. But we didn't really know very much about them intrinsically. Anyway, the field thought, well, we got it all now. We don't need the assays anymore. We'll just isolate the cells, and we will understand them by analyzing them directly, because when you do the functional assay, you don't have the cell anymore. So you could purify these cells by flow. You could look at their RNA, their DNA, their proteins, and you could manipulate them short term and understand how they're regulated. We were a little skeptical of that, even though we thought we would start with the most interesting cell, because our lab had also been working on the mouse system where we could work actually with single cell transplants and work with highly 50% pure stem cells. And we had discovered that through serial transplants of single clonally analyzed cells, that they're not all multipotent. And not only that, you could put them in vitro in short-term scenarios and show that the control of their survival, their proliferation, and their self-renewal, their generation of daughter cells through a division or two, can be differently regulated. So you could have this without, you could have this without this, you could have this without this, et cetera, et cetera. So, so David Knapp comes to the lab and he wants to study those primitive cells molecularly. And I said, whoa, just a minute. Let's just do single cell analyses of these cells and find out how homogeneous they are, because if you're going to analyze them molecularly, you better know that. So he, and then later joined with Colin Hammond, who's actually still doing his PhD, isolated this complicated phenotype I described. Oh, here it is up here. Index sorting, okay? So you know exactly how much antigen of every type of, using every type of antibody up here, is expressed on the cell surface. And you can see they're not all identical, there are a bunch of them. So they indexed single cells into wells and transduced each one with a different one of 57 barcoded vectors that we had developed. And then they could combine all 57 and put them into a single mouse. So in 12 mice, they could analyze 684 cells. And Fang Wu, who actually just in her third year PhD, first thing she did and come into the lab was modify that culture system that Heather had developed so that it would support the growth not only of granulocyte and macrophage clonogenic cells and terminally mature cells, but also members of the lymphoid compartment. So, you know, looking at multipotency from that perspective. These guys got 7% of all the cells read out clones, all of the transduced cells. Fang Wu's culture read out 82% of the cells, didn't need barcoding. So we knew the cells were viable, and we knew the cells would yield us information. And here it is summarized. You can see every line is a clone. Every color is a different kind of clone. These are the ones that are in your papers. These are the predominant ones. And you can see the spectrum of readouts. Sort of similar, actually, to the mouse system. And lo and behold, here's the results from Fang Wu's culture. In 6 weeks, she reads out what the guys did in 30 weeks in the mice, remarkably similar. So we kind of think maybe these cells are predisposed to do different things in terms of their differentiation. In other words, their epigenomes are already jiggled a little bit, and they're not irreversible, but that's kind of what they're going to do on both of these circumstances. So then, of course, David Knapp and Colin and Feng, we went ahead and did the molecular studies. And the bottom line is the protein level, at the RNA level and at the epigenetic level, the methylome level, all of which done on single cells, all of which index sorted. It's a mess. You can see it's very heterogeneous. Now, some of that may be reversible heterogeneity, but it tells us that these cells are like cities, you know, they're not all the same, even though they have all roads and stop signs and people moving around. We have a lot to learn still about how to understand them and make predictions about what they're going to do. So now what about AML? Well, we didn't want to study this. Everybody's doing this, and it's a mess. I don't really think we're going to understand leukemia or other cancers by just trying to sample a little bit of what is a huge mess of stuff. It's not new to think about going back and do a prospective analysis. In fact, that's what the transgenic mice people have been doing for years. But we wanted to do it with human cells because we don't think mice are close enough to really make the kind of predictions that we need to treat human cancers. And that's not a new idea either, but it's been extremely difficult. In fact, John tried that in the 80s, and to this day, there have been only 2 or 3 systems that did that. So Alan comes along and he says, well, come on, kiddo. Why don't you try chronic myeloid leukemia, chronic phase cells? So we spent the whole of the 70s and 80s studying chronic phase CML. And back in those days, before Gleevec, we knew that patients would progress from chronic phase into blast crisis. So the idea was you'd get these cells, which we still had some in our freezers, and push them over, and that would be a whole lot easier than starting way back here. So the question was, what genes? So here's Keith Humphreys up here. Keith Humphreys was my first PhD student. So all of you who are junior PIs, make sure your first student is the most brilliant person, better than you, and your career will be wonderful thereafter. Because you got to marry well too. So Keith pioneered understanding the role of Hox genes in hematopoiesis and also played a role in illuminating the fact that a lot of Hox genes, particularly when they're connected to NUP98, are active in leukemia. So that was, and he had made vectors with various versions of them, which he let us have. So here's the experiment. We have in our freezers the primitive cells that make this chronic phase disease when differentiation is not altered, but their ability of the stem cells to maintain themselves, which is one of the things that Alan and I discovered, is compromised. So we're going to try and push that. So the cells would go from here to here, or to ideally to here, and see how that works. So we tried Keith's vectors, and we got the cells to go with this vector that expands stem cells to go from here to kind of expansion, but not into these compartments. And here's NUP98 HOXA9, which is in acute myeloid leukemia. We could take these cells and we could push them beyond accelerated phase, but not quite into a real full-blown blast crisis. And Philip Beer was screening new genes because of these being a bit disappointing. A whole bunch. He made a whole bunch of vectors, including NA9 he tested in his in vitro screen, and IK6, which is a construct that suppresses Ikaros, because Philip thought he was going to make acute lymphoid leukemia, and suppressed or mutated Ikaros is common in that disease. And that actually took the cells right into accelerated phase and allowed us to discover that, in fact, in patients, accelerated phase, every patient we've looked at, quite a lot, a large number, have suppressed Ikaros. Obviously not how we manipulated it, and we don't understand that, but it's interesting that that has some clinical relevance. So the other gene that was positive in the screen was MYC. Heaven forbid that I should work on MYC. The whole world works on MYC, and I don't know anything about it. And here's Lisa Believa, a new student working with Philip, and she says, I want to do that. And I said, no way. We're running out of CML cells that have predominant chronic phase CML stem cells. Go do something with normal cells and show me that you know what you're doing. So she did that. She took some cord blood, put MYC in it, showed it was overexpressed a bit. So I said, yes, you can put them in some mice. Long story short, first experiment, she comes back with mice dying in somewhere between 3 and 6 weeks, all of them, and they have full-blown leukemia. So she used as her first mice that are engineered. Keith and I developed these mice back in the early 1980s and then crossed them forward as we got better and better immunodeficient mice. These mice constitutively express human IL-3, human GM-CSF, and human Gland or stem cell factor, because we knew already at the time that these two, IL-3 and GM-CSF, the mouse factor does not work on the human receptor, and leukemias like those factors. So, and in fact, it's been found since then that most leukemias grow better in mice that are expressing these because they're now available worldwide through Jackson. So then, of course, we thought maybe just put it in the control mice because not everybody has these and they're going to think, you know, Eaves has gone and done something crazy again. So she puts them in these mice and no leukemia, no nothing. Mice live, they're happy. So then we thought we better look at what's going on. I thought the mixed cells would all have disappeared. No. These mixed cells, because we put into the mice both the ones that got MYC and the ones that didn't, they do just as well, okay? The mixed cells are the solids, the non-transduced cells are the open ones. Every single kind of cell that grows in these mice, produced from these cells, is produced from these mixed cells, like they think they're perfectly normal. So then, of course, we thought, well, we'll do secondary and see what happens. And of course, normal cells don't do very well in a secondary transplant for anybody, and nor did these, or I should say these probably. We just put the whole bone marrow in. But you put them into the NRG3GS mice, and bango, they get leukemia right away. So these cells are sitting there perfectly normal, behaving perfectly normal. They haven't done anything. You just trigger them with inflammatory growth factors, and they get leukemia. Isn't that scary? Doesn't that make you think it's not just mutations? And the other tenet was you've got to start with a primitive cell. So Lisa was stimulated to put this MYC gene into the latest possible granulocyte macrophage precursor that will go in a mouse and do anything, and bango. Very efficient. Look at the efficiency of leukemia from a limiting dilution. In fact, she's done single cell transplants. And these are serially transplantable. Here's the late guy making AML, secondaries, tertiaries. Ugly. So breast. Got a few minutes left. I'll tell you about breast. Why did we get started in breast? Because Shaki Dedhar came to me in the late 80s. He was studying cell lines, solid cancer cell lines, response to growth factors, and he wanted some controls. So he heard we had growth factors for hematopoietic cells, which everybody knew they were very specific. So I said, sure. And then he comes back and he says, I have some of your growth factors stimulate breast cancer cell lines. And at that very same time, Bill Peters at Duke University had just published a paper saying, we're going to cure breast cancer with bone marrow transplants. And by that time, transplant recipients were already getting hematopoietic growth factors to stimulate neutrophil recovery. So we thought, oh my goodness, maybe they're going to stimulate the breast cancer. That won't be a good thing. And Alan says, go away and do something important because you're supposed to be helping the bone marrow transplant program. So I thought, well, cell lines, we always dubious about cell lines. I'll go and talk to somebody who really knows how to work with primary human mammary cells. That turned out to be Joanne Emmerman, who is Canadian with a lab at UBC, back from Mina Bissel's lab in California, where she trained and learned how to do this. So we took the growth factors out. She said, sure, add them. We added them. Did nothing. She thought she was growing breast cancer. Actually, now we know she wasn't. She was just growing out the normal cells. But it didn't do anything. But I saw, and then Bill Peters' thing got shown to be not really what he made it out to be, so the whole thing went aside. But I saw how these cells grow. You know, I never thought about breast like skin and gut and everything else. I thought, well, it's got to have progenitors, make colonies, stem cells. And Joanne said, oh, no, you're crazy. You know, they have to hug each other and sit on a basement membrane. That will never work. But she had a student, John Stingle, who was about to start his PhD, and he said, I think that would be very cool. So we worked, she said I could co-supervise him. So we worked out a system with stroma, actually, to do it at limit dilution, or now make colonies. Long story short, along with Afshin, who helped optimize the system, you can make colonies of luminal cells. Those are the cells on the inside of the duct. You can make pure basal cells that are myoepithelial in capacity. And you can make mixed colonies. You could put single purified cells in a well, and some of them would make a mixed colony with luminal cells in the middle and myoepithelial, because they migrate on the outside. So, on to stem cells. So John had heard that you could grow mouse mammary cells in the fat pad of a mouse into which the mammary gland was just coming in the 3-week-old baby female mouse, and you could cut that off and then use that as a sort of transplant scenario. So you could eject cells and make a new mammary gland. So he purified them, learned how to purify them, and could even do a single cell. Here's a single cell reconstituting a mammary gland. That was great. So let's do it with human cells, I said. I don't want to work with mice. Well, it turns out human cells don't like fat. They're different. You know, most mammary likes fat all hugging around it. Human mammary cells, the mammary gland likes fibroblasts all around it. So okay, we'll put them in collagen gel with a lot of fibroblasts so we can do limiting dilution assay. And yes, you can reconstitute a human mammary structure in this little collagen gel that we originally put under the Kids capsule, but it works. You can just put it subcutaneously. And the cells, you can't tell them apart from a human mammary gland, and they have basal cells on the outside and luminal cells on the inside. You even make milk if you get the mouse pregnant. But that was not an assay. So Peter Iru comes along. He's a physicist. He likes mathematical things, and he remembered Heather's long-term culture assays. So he said, let's digest this, just like a mammary gland, and assay for colony-forming cells. And that turns out to be a perfectly quantitative assay. And you can do self-renewal. So then along comes Long and says, I want to do the same thing as you guys did with leukemia. I want to do breast cancer. And I said, you're crazy. You'll never do that. Nobody's made a solid tumor out of primary human cells. But he went to the freezer and got Philip Beer's vectors, and Philip happened to have the ones that are the most commonly involved in breast cancer in people, except for KRAS, which affects the pathway which is affected genetically. And when she, just like Lisa, first experiment, he gets a tumor. It's a bona fide tumor, can be serially transplanted. And the clones in the secondary tumor are different from the first. But it's a pathetic tumor. After the first 4 to 6 weeks, it doesn't grow anymore. It stays there, it's turning over, makes secondaries, but they do the same thing. So subsequent people in the lab, Sylvain, started trying a lot of different vectors. And he discovered with mere AKT alone, you could actually make DCIS. So this is not as good as KRAS, right? But it's a very interesting early stage cancer. Looks just like DCIS in people. And here is Susanna Tan. She's just about to take her comp in a week or two. And she's now got these humongous tumors. These tumors are so big with KRAS plus 3 other oncogenes that the mouse colony people make us sacrifice the mice. And they serially transplant. And this is all very new, but I'm presenting it because I think we are on the verge of a new era. We are in a new era where we need to change our thinking. And this is just a summary of how we need to change our thinking and how we're going to be able to change our thinking with these new ways of isolating cells, analyzing cells, thinking quantitatively, and doing things that we never thought we could do before. So like everybody else, I want to close by thanking, these are all the people that we've trained in our lab. These are the kids that are there now. I was given this as a birthday present by my lab when I was 70, and I've kept upgrading it. And before I end, I would just like to acknowledge not just the collaborators, but there's a whole army of people backstage who really help us scientists be scientists. And they deserve to be recognized, the technicians, the project managers, the administrative staff, and the support people in the specialized facilities. Thank you.